Hi everyone, I'm Zixia Chen from CSBA. The paper I will be presenting for today's journal club is a preprint recently published on bioRxiv titled Metabolically Robust Autoluminescent Reporters. Autoluminescence technology has demonstrated immense potential in vivo imaging because it requires no external excitation light and has extremely low background noise. However, it has historically been constrained by metabolic interference, making it difficult to utilize for precise quantification. This paper addresses and solves this issue. Figure 1a illustrates the fungal bioluminescence pathway. Its ingenuity lies in its close connection to the host's natural phenylpropanoid metabolism. Intracellular caffeic acid is converted into fungal luciferin by the enzymes HISPS and H3H. After the luciferase catalyzes the light emission, the oxidized product is hydrolyzed and recycled back to the caffeic acid by CpH. This closed-loop substrate cycle enables continuous luminescence without the need for the external addition of substrates. Directly expressing fungal enzymes in plants or other heterologous hosts often lead to disappointingly dim luminescence. To fix this, the authors published an optimized version, NN-Luz V4, in Natural Methods in 2024, which massively improved brightness in eukaryotes. This V4 version actually served as a baseline for the initial mutant library screening in this current study. But as the paper was coming together in its final stages, the team swapped in NN-Luz V5, a brand new, much more powerful version they had just developed in a parallel project. By adopting V5 as a co-luminescent engine, they were able to push the boundaries of brightness even further. Moving beyond the fungal system, Figure 1b highlights another classic route, the bacterial bioluminescence pathway, specifically the Lux operon. This system relies on reducing equivalent and long-chain fatty aldehyde. Working with this pathway, Kusuma's groups used the BRET, bioluminescent resonance energy transfer, to fuse fluorescent proteins directly to the bacterial luciferase Lux A, successfully creating multicolor luminescent variants. That previous study is crucial because it laid the physical groundwork for the paper we are looking at today, proving that it's entirely feasible to shift the autoluminescence emission spectrum using BRET. Even with these luminescent tools in hand, when we actually try to build biosensors, we run into a metabolic trap. If a certain patch on a leaf grows incredibly bright, is it because of the target hormone level is actually high, or is it simply because that the specific region is metabolically hyperactive, flooding the system with fuel like caffeic acid? A single-color autoluminescent system simply has no way to distinguish between the two. Because of this, trying to quantify absolute signal intensity in vivo is extremely susceptible to background metabolic noise. To completely break this coupling, the authors outline their co-design strategy. They co-express two different luciferases with the same cell. Because they are in the same environment, they compete for the exact same pool of metabolic substrates, but they emit different colors of light. The green signal is driven by a promoter responsive to a specific physiological cue, while the red signal acts as an internal control, driven steadily by a constitutive promoter. By simply taking the ratio of the two, any substrate fluctuations caused by background metabolism are mathematically cancelled out in the numerator and denominator. What are left with is a pure quantitative, isolated readout of the physiological signal. The fungal pathway is natively green, but red shifting requires an integrated fluorescent protein. Since BRET efficiency scales inversely with the power of the distance, surface fusion is ineffective due to the resulting gap. The authors instead chose to go inside the enzyme to find structural safe room for the acceptor protein. They performed a systematic sliding scan using a 17 amino acid probe, shifting every 3 amino acids. This two-in-one strategy utilized MLY1 and BTGZ1 sites to test tolerance for 17 amino acid insertions and 2 amino acid deletions. This scanning allowed them to pinpoint internal sites like 119, 197, and 188 that tolerate structure perturbation without sacrificing enzymatic function, paving the way for our redshifted variants. Initially, a single internal insertion impaired the enzyme structure and severely reduced the brightness. To fix this, the authors employed a dual antenna strategy by fusing a second copy to the tail. The internal copy provides strong close-range absorption, while the terminal copy catches residual green light, remarkably restoring both stability and absolute brightness. Applying this strategy yields the final variants in Figure 2b. Note that the M-scarlet I3 delta C10 tag in the architecture. To minimize the distance to the luminescent center, they truncated 10 flexible amino acids from M-scarlet tail to remove steric hindrance. This meticulous fine-tuning ultimately generated the highly efficient redshifted variants Luz 593 and Luz 608. To demonstrate that the ratiometric approach is a universal physical principle rather than just a fungal-specific phenomenon, the authors incorporated another tool into their experimental system, the BRAT-engineered multicolor bacteria luminescence variants, as shown on the left in Figure 2c, from Kusuma's 2024 paper, which proved that this multicolor bacteria system can directly generate bright and distinct autoluminescent implant host, specific in Nicotiana bassamiana leaves. This high level of in vivo compatibility in plants established a solid foundation for the subsequent parallel validation of metabolic robustness across both systems in plant leaves. In Figures 3a and 3b, the authors deliberately induced severe metabolic perturbation by applying a high dose of 1 millimole TDZ or by alternating oxygen availability. Under these conditions, the absolute luminescence intensity of a single color fluctuates widely. Relying solely on single color data would be completely misleading. However, by taking the ratio, these metabolic shifts are neutralized. In the bacterial system, this ratiometric result was up to 16-fold more stable than the intensity-based measurement. The authors next calibrated the system by treating BY2 suspension cells and leaving tobacco leaves with varying concentration gradients of TTZ. The results clearly show that the absolute brightness of individual colors suffers from severe metabolic delay over time. However, the calculated ratio not only stays completely stable but also displays a perfect stepwise dose response. By extracting this steady ratio during the plateau phase, we can plot a precise standard curve, entirely upgrading the measurements from unreliable qualitative bright or dark data to absolute qualitative homo concentration. This slide demonstrates the system's powerful spatial temporal denoising capabilities. The left image shows the long-term temporal stability. Using the WRKY70 promoter for salicylic acid biosensor, the ratiometric system successfully figured out micro-metabolic fluctuations over a weeks-long developmental cycle, maintaining baseline stability in the absence of salicylic acid-inducing stimuli. The right image shows a spatial denoising comparison. when triggering a genomic acid response via Pseudomonas savantae infection. Traditional single-color imaging highlights nutrient-rich leaf veins, a classic metabolic artifact. However, the ratiometric map completely eliminates this false positive, precisely localizing the true immune response on a 2D plane. So, in this paper, they introduced a new approach, co-expression enzymes that use the same substrate but emit different colors of light. This effectively removes metabolic background noise. Second, they successfully redshifted the fungal enzymes using a creative strategy, inserting sequences internally and adding double copies at the tail. Third, by using a standard curve, they transformed an easily disrupted qualitative method into a highly reliable quantitative tool. This new system strongly resists interference in both healthy and diseased live models. That's all. Thanks for your listening.